Assalamualaikum warahmatullahi wabarakatuh. My name is Fatika Sari. I am from Biologi A 2020. My student number is 4411420004. Today, I would like to share the presentation about my publication journal. The title is Microbiological Observation to Identify Bacterial Groups from of Bacillus subtilis and Escherichia coli by gram staining method. The author is me, Fatika Sari. My study program in Biology, Faculty of Mathematics and Natural Science, the institution in Universitas Negeri Semarang. And then this is my email address. The first time I will start my presentation from abstract. Bacteria are a type of microorganism that cannot be seen without the aid of a microscope. Prokaryotic bacteria are a generally single-cell or unicellular, have a very small size, even smaller than protozoa and fungi. The cells are colorless, making it difficult to observe under a microscope. The aim of this research is to identify groups and characteristics of bacteria by performing gram staining using the bacteria Bacillus subtilis and Escherichia coli. Gram staining method using several dye reagents including crystal violet, iodine, acetone alcohol, and safranin. The bacteria used in this research is Bacillus subtilis and Escherichia coli. From this method, bacteria can be classified into groups, namely gram-positive bacteria and gram-negative bacteria. The resulting color is an indicator of the two types of gram bacteria. The gram staining of Bacillus subtilis gives a purple color which shows the characteristic of gram-positive bacteria, while in Escherichia coli show a red color which is a characteristic of gram-negative bacteria. The benefit of this research is making observation in the field of microbiology to identify gram bacteria, and the reader can see the difference in the shape, color, characteristic of bacteria as a result of gram staining. The keyword is gram staining, gram-positive bacteria, gram-negative bacteria, and the cell wall. My introduction contains that bacteria cells are generally colorless and difficult to observe under a microscope because the cell can show a color contrast to the surrounding medium. Bacillus subtilis and Escherichia coli are two of several pathogenic bacteria. Gram staining is most widely used in the research because it is easiest to identify the type of bacteria. Gram staining cannot be done on microorganisms that do not have a cell wall. In the field of microbiology, bacterial staining aims to make it easier to see bacteria when observed under a microscope, can clarify the size and shape, show the presence of cell structures such as the presence of cell pores and granules, and be able to increase the contrast of microorganisms with their cells. This study aims to look at characteristic bacterial cells and easily divide bacteria into groups, namely gram-positive and gram-negative. This research is able to provide benefits to students in making observation in the field of microbiology to identify gram bacteria of Bacillus subtilis and Escherichia coli. And then the readers can see the difference in the shape, color, and characteristic of bacteria as a result of gram staining. Material and method. The material used in this study were cultures of Bacillus subtilis, Escherichia coli, for dyes reagents including baker crystal violet, lugol, alcohol acetone, safranin, and immersion oil to have observed under microscope. The tools used are object glass, cover glass, Bunsen, microscope, dropper pipette, loop, flat top, flat dryer, and clamp. Gram staining method is adding crystal violet, lugol, alcohol acetone, and safranin on bacterial sample. This research was carried aseptically using 70% alcohol solution and Bunsen flame to avoid contamination with other microorganisms or remain steril. The first step in the gram staining method is the slide was cleaned with alcohol and sterilized on a Bunsen. And then when loop of Bacillus subtilis and one loop of Escherichia coli were taken, which then placed on a slide and being air dried. The slide was fixed on a Bunsen flame and cooled again, and then drop of reagent crystal violet on the bacteria sample and stir for 1 minute. After that, it was with running water and dried. And then the second dye was dropped on the stand, namely iodine. Left for 1 minute, then was with running water and dried. The slide containing the bacteria culture was the washed again with acetone alcohol from approximately 30 seconds, then was with running water and dried again. And then the last step of gram staining is to incubate the bacterial sample with safranin reagent on the for 1 until 2 minutes, and then wash it with running water and dry it on the slide dryer. The slide were dried with immersion oil and observed under a microscope. Result and discussion. Bacillus subtilis done with the gram stain method showed a purple or violet color, while Escherichia coli produced a red color. This indicates that Bacillus subtilis is a gram-positive bacteria and Escherichia coli is a gram-negative bacteria. In the picture one is a gram-positive stain colonies of Bacillus subtilis, and then in the picture two is a gram-negative stain colonies of Escherichia coli. Gram-positive bacteria are bacteria that are able to maintain the basic color of crystal violet during the gram staining process so that the basic color is visible when viewed under microscope, even though the alcohol acetone washing process has been carried out. Gram-positive bacteria have a thick peptidoglycan walls and little lipid appear blue or purple because they retain resistant crystalline violet, and then disease of bacteria will be blue or purple under a microscope. On gram staining with Bacillus subtilis, the color of the bacterial cell is blue or violet. In addition to the gram color produced from this staining, it can also be seen that the structure of Bacillus has the shape of bacil. Some are colonies or solitary. And this is the gram-negative bacteria. I explained that the gram-negative bacteria that is not able to maintain the basic color of crystal violet during the gram staining process after going through the washing process with acetone alcohol so that it will run when observed under microscope due to the covering dye by safranin. Gram-negative bacteria are having a thin cell wall, a triple or multi-layer cell wall membrane, having a thin peptidoglycan layer but containing a lot of lipids, not containing teichoic acid and less susceptible to penicillin. Gram-negative bacteria have a cell wall structure with lipid content so that it will be washed off by alcohol. Gram-negative bacteria have one layer of peptidoglycan so that they will be destroyed by the decolorization of acetone alcohol, which causes the purple crystalline dye to come out during the gram staining method. Cell wall gram of bacteria, gram-positive bacteria, which have a thick net-like cell wall consisting of peptidoglycan, which is purple in color. Peptidoglycan is mainly a polysaccharide consisting of two subunits called an acetylglucosamine and acetylmorocanic acid. When adjacent layer of peptidoglycan are performed, they are closely linked by short peptide chain via the enzyme transpeptidase, resulting in the shape and rigidity of the cell wall. The tight peptidoglycan layer of gram-positive organism allow this organism to retain the UV iodine complex and stain the cell purple. And then in a gram-negative bacteria, which has a thinner peptidoglycan layer and losses crystal violet iodine complex during the colorization by alcohol rinsing, but retain the same opposite safranin. So it is apparent that these are pink. They also have a additional outer membrane which contain lipids which is separated from the cell wall via the perisplanchnic space. Again, it is for the reagent of gram staining. It is have several functions. Crystal violet serve a basic or primary colors that gives color to the bacteria want to stain. And then the goal is the function to increase the activity or increasing the dye stuff, clarifying the basic color and inhibiting the solubility of the dye. Acetal alcohol serve to pale the color of the purple crystal on the glass preparation that have been attached to the bacterial stem, and then safranin give bacteria a color that can be stained with crystal violet. The conclusion of this research is can be concluded that Bacillus subtilis is gram-positive bacteria, while Escherichia coli is a gram-negative bacteria. This is my reference for this research. That is all for me. I would like to say thank you, and I apologize if there any mistake. Assalamualaikum warahmatullahi wabarakatuh.